on these topics. So, and I just have to look over and make sure that like.
Okay. Patrick, can I see your face again? Can I get up?
And I just know these.
Here he is. I didn't want to ask anything. I just think you're a good looking guy. I want to see your face. No, I just wanted to make sure you could hear me. So let me see. I don't know if I have a camera working now.
I can hear you. I'm not sure if you can hear me, but I can hear you.
I can hear you. Yes, good. So, for some reason my camera isn't working, but I think you can see my screen, and that's really what's important, right?
We can't see your screen yet.
Okay. Okay, how's that?
Yeah, a large version of myself.
Okay. Okay. All right, so here we are. We just had a wonderful break. Oh.
We can see.
You even know how to do that. I got this to it. No? Come on, man. How do you forget?
Right there.
Is that this guy?
Yeah, maybe. Power button.
There we go. Thank you. Okay, rock and roll. All right, we're in business, people. All right. So real quick before we get started, Dr. Lounsbury talked to me last week. She said a lot of people were freaked out by that announcement I sent out. That wasn't for you. It was for my students in Plattsburgh. And everybody's like, oh my God, who's this guy? He's so mean. It's like, let me explain to you what that was. So let me show you the exam I gave at Plattsburgh. So this is the results of that exam. So 45% of people got an A or B. They're okay. They're actually taking the class. 55% basically didn't learn anything in 3 weeks. Okay. So they're not coming to class. They're not studying. They're not doing anything. And I'm like, these kids need a kick in the ass. So I gave them a kick in the butt. And I'm telling you, last week, they were sitting in the front of the room. They were asking questions. It's like, hello, now you're students. So sorry you guys had to deal with that, but sometimes I need to give people a kick in the butt. I didn't sense that any of you need a kick in the butt, though. I'm liking that about you people. No kicks and butts.
Are those students undergrads?
Yeah, they're undergrads. Yeah. And it's been hard ever since COVID. It seems like the quality of students has really gone down. And I think a lot of these kids would be better off. They took a couple of years off and worked, figure out what they want to do for a living, and come back to college because they're just kind of, you know, getting stoned and skipping class. You know, it's not a path forward. But anyway, that's not your problem. So look, here's what your problem is. No, actually, you guys did pretty well in the last exam. I'm going to show you the results first, and then we'll go over the exam. Class average is 87. So I added 8 points on everyone's exam score, and that's included here. You won't see it in your score on Brightspace in underneath the exam itself. I put them into the bonus points. So there's that. So let's go over the exam just so that we all know what we're talking about here. Let me do this. Preview. This seems like 100 years ago, doesn't it? Okay, which histamine receptor is the target for stomach acid reducing drug omeprazole? Should have known that was going to be on there because I'm such a big fan of it. It's what allows me to live a normal life. Which kinase is activated by NO? That's protein kinase G, makes cyclic GMP. Which of the following pathways is not associated with IP3 calcium? Wnt has got a non-canonical pathway. This has got a pathway to it. This definitely is. Hedgehog is the only one that doesn't really activate that. Right? This is your phospholipase. There's too many C's in this, but this is your phospholipase C activated by alpha Q, and then the RTK's got your protein kinase C gamma, as does the Wnt. No, the Wnt is the same as this one. But anyway, lipophilic hormone made from vitamin A, that is retinoic acid. The order of events: B cell encounters antigen, B cell produces IgE. See, it's easier when they're in the right order. Okay, so the B cell then makes your IgE. The IgE binds to the mast cells, and when that antigen shows up again, they degranulate. Which local modulator is not made by COX? That is leukotrienes. So these are all part of that COX pathway, and leukotrienes are not. Protein that helps to recycle elements of the vesicle fusion, the NSF. NSF and the SNAPs take the snares apart so that the V snare can be recycled. Which of these signaling molecules has a pH domain? mTOR, GSK does have it. Oh, definitely mTOR. RAB2 does not. PI3 kinase does not. Neither does GSK3 beta. Okay. In addition to that, so I haven't looked at these questions in probably 3 weeks. In addition to the activating cell division, AKT blocks apoptosis. Which target of AKT is resulting in less apoptosis? That would be, oh my God. Oh, this is related to translation, GSK3 betas, cell division, mTOR. It's not FOXO. FOXO is going to activate BIM, production of BIM, and we'll talk about BCL2 proteins today. So BIM is a pro-apoptotic BCL2. So FOXO is the answer there. Which transcription factor has both SH2 binding domains and SH2 domains and binding domains? Those are your stats. Okay. Epinephrine is hormone released by the adrenal medulla in times of stress. It is carried by the blood to all cells outside the CNS. Not all cells respond the same way. So this is kind of the way we started that chapter. Basically, a cell's not going to respond unless there's a receptor there for the hormone. Number two, it depends on which subtype of the receptor is on that particular cell, what the response is going to be. And number three, depends on what that subtype is going to be linked to, which G protein is going to be linked to produce the response. So you've got variability at all those layers.
I was also thinking like it might be concentration dependent as well, like how much is presented within the, because that's
Concentration of epinephrine?
in terms of like either receptor or like how much epinephrine is presented. Well,
I don't know about that. Yeah, I'm not following that one. I'm not really sure. Yeah, sorry. Any other questions? There are two different types of G proteins in cells and many isoforms of each. The two main types are small monomeric and large heterotrimeric. So what's the difference? So they all bind GTP and they're activated by having GTP attached. They both hydrolyze GTP, although the small monomerics need GAP to help hydrolyze it, whereas the large heterotrimerics will break it down themselves. How else are they different? The small monomerics tend to be linked to cell crawling, which is rearrangement of the cytoskeleton, and they are linked to cell division and trophic factors and stuff like that. Whereas the large heterotrimerics are more physiological. They change cell physiology and they're very rapidly reversible. So they could be linked to usually increases in cyclic AMP or IP3 calcium. So they're two very different pathways there. Questions about that? Okay. P10 and GAP are proteins that cause cancer when they're not present rather than they're present. Why is this? Because these are negative regulators. So GAP will turn off small monomeric G proteins. P10 dephosphorylates PIP3 back into PIP2. So these can cause cancer because if they're not present, then those pathways stay on longer than they should, and they cause the cell to continue to divide. A general mechanism that's commonly used in signal transduction is recruitment. I'm a big fan of recruitment. This is when one activated protein brings other proteins into the complex. Give an example and explain how this works. So the key is we're bringing proteins together, and that puts the enzymes near their substrates. So for example, you activate an RTK, you pull up PI3 kinase. Now the PI3 kinase is next to PIP2, and it can phosphorylate that, make PIP3. Or you bring SOS and GRAB up to the membrane. Now it's near RAS, which is membrane attached by a lipid, and so you can activate RAS. So it's all about bringing the enzymes close to their substrates, and that turns on the pathway. I'm going kind of fast. I hope that's okay. It's just that this was so long ago, and I kind of want to get to the new stuff. Okay, a research chemist develops a new drug that has no effect on postsynaptic norepinephrine receptors, but enhances neurotransmission through norepinephrine releasing receptors, two mechanisms. So basically, we're talking about the presynaptic axon bulb and modulating that to make more norepinephrine in the synapse. Most obvious, block the reuptake, right? Obvious one is block or activate, block the presynaptic autoreceptor. Both of those are going to leave more norepinephrine in. Other mechanisms are things like blocking the breakdown of norepinephrine so that it gets, so more of it stays in the synapse, more of it gets pumped into the vesicles, and then more gets released. That's how your tricyclic antidepressants work. Also, it could be a precursor to the norepinephrine, like what is that? L-DOPA. L-DOPA is used for Alzheimer's patients. L-DOPA gets converted into dopamine, which gets converted into norepinephrine. So that's going to increase norepinephrine in the synapses. I heard a podcast once about a woman who had Parkinson's. They gave her L-DOPA, and she suddenly developed this gambling addiction, and she lost hundreds of thousands of dollars because L-DOPA, dopamine is important for movement, but it's also important for addiction. And she got addicted to gambling based on her L-DOPA. It just gave her more pleasure gambling than it would a normal person because there was more dopamine in her whatever. All right. You got any other answers here you want me to discuss, or is that good? Is that good? Okay. All right. So like I say, you guys did really well on this exam. Again, I have to keep making these exams harder because you guys are so smart. Should I or no? No?
No,
No.
that's not ours, man. But did you end up deciding what you're doing about the next exam?
Yeah, I only heard back from about 4 people, and they're all very positive about it. So I assume we'll go ahead and have the next exam next week.
Yeah.
Like from Tuesday through Sunday. That's usually what we do. It gives you plenty of time to study and then take the exam. The reason I did that is because I started in looking at, like, I was going to go straight from cell division into cancer. And I started writing cancer notes, and it's a lot. I mean, cancer's a lot. So I don't want to throw that on top of this. And then you've got so much material and only, 10 multiple choice and 5. It's not going to adequately judge how well you know the material. So I think it's just going to work out better for us. So what I want to do here today is review the stuff we did the last time we had class, and then go through apoptosis. And that should be enough for this exam. And then Thursday we'll get together, we'll review the whole chapter. And then today I'm going to put together a practice exam for you, 5 of the more complicated questions, the written questions. So if you can look them over before Thursday we can talk about them. and you can get an idea of how I think about answers to these questions. And I'll help you to focus on how to answer these questions. Okay, sound good? That's what we're gonna do from here out. Yep.
How many questions, like multiple choice, do you think will be on this next one? Like
How-
similar to the
I-
previous exam?
Yeah, same as the previous exam, 10 multiple choice and 5 written answers. Does that seem good, or do you, yeah? See, the second exam, I wanted to do more multiple choice, but I thought, well, you're already ready for 10 multiple choice. And if I change the exam suddenly, it's gonna be a hard thing for you guys, so I left it the same, if that's okay. Okay. Sometimes I worry about that, because like multiple choice, sometimes it can go 50-50. And you miss one multiple choice, that's 5 points. It's a lot, you know? So, yeah, I think we'll stick with 10. Yeah. All right, what else do we got? Any other housekeeping here? I guess not. All right, so let's go back to what we did last time. So in the first couple lectures, we talked about cyclins and in terms of this G2 to M transition. And we talked about cyclin B and CDC1 or CDK2. CDK2? CDC1 or CDK1. CDC2, sorry. CDC2 or CDK1, which is our mitotic CDK. And they come together, and then you've got that series of phosphorylation, dephosphorylation events that gives you a really rapid transition into mitosis. And then you get activation of APC when all the chromosomes are lined up on the microtubules have attached to all the kinetochores, releasing CDC20, activating APC, degrading that cyclin B and getting out of mitosis. So we talked about that previously. So in the most recent class, we were talking about this, our transition from either G0, a rest state, or continuing in the cell cycle. If it's a stem cell, that's going to divide anyway. What does it take to get through this restriction point, which is R? And we're going to go back all the way to the last chapter with signal transduction. So again, RTK, RAS, RAF, MEK, MAP kinase, activating some transcription factors. So we're phosphorylating transcription factors, and that's activating transcription. Now, the first set of genes that get transcribed are IEGs. And keep in mind that you don't need protein synthesis to get these IEGs transcribed. Everything you need, this whole chain of pathways there until you get to the IEGs. Now, to get to the next step, these IEGs are going to be transported out of the nucleus, come back in, and a lot of them are transcription factors. And then we're getting these, E2F, cyclins, and CDKs. And here is an experiment showing that. So here you've got a bunch of serum-starved cells that when you starve them of serum, they all get stuck at the in G1. So then you hit them with a serum, which has got platelet-derived growth factor in there, activate RTKs, and you get this whole series of genes being turned on. The red ones are on, the blue ones are off, or low anyway. So you get this series of genes turned on 30 minutes after you're exposed. Then after those go out and get transcribed, they come back in and you get other genes being turned on. These are your DEGs, delayed early genes. And then those turn on a third set of genes, which are going to be your S phase genes here. So again, question.
How is IEG and DEG different or similar in terms of like, because it seemed like it goes through IEG and then it goes into DEG.
Right. They're different proteins. So when you first hit with the growth factor, you get specifically more Jun and MYC. MYC is key. We'll talk about MYC today. And MYC and Jun Phos are going to turn on that second wave. So the first set of transcription factors get activated, make other transcription factors. Those transcription factors come back in the nucleus and do a whole different set of genes. And those come back into the nucleus and turn on a whole different set of genes. So this is gene cascade where transcription factors are activating transcription factors, or activating transcription factors. Okay. Interesting thing about that, I got in, you know how profitable it is to get into an argument on the internet. I got in an argument on the internet once with an older developmental biologist, and we're talking about how a small change in one gene could cause a large change in an animal. And he's like, well, how could one protein cause a huge change? Well, if it's a transcription factor, that's like a master switch. You change that, you change everything down the stream from that. You have massive changes. So this gene cascade is really important, and everything has to go right, basically, or else things change. Okay. All right. So 2 waves of gene transcription. And then here, DEGs, this is the key. So we've got cyclins and CDKs, but the key is E2F. E2F is the transcription factor that's going to get us past the restriction point and into the cell cycle. And as soon as it's made, it's going to be grabbed by RB. And we'll see in a minute, RB brings in a histone deacetylase, histone deacetylase, and that causes the chromatin to condense. And then you get phosphorylation of RB by the cyclin CDK, that releases E2F's activity, and you go into S phase. That's kind of like the Bio 101 version of it. It's more complicated than that, though. And here we start to talk about how this cell cycle clock. So the clock is always going in the forward direction because of different cyclins are needed to get you through from one point to another. And then that cyclin is degraded, another off from it. So I kind of like this view. This is the same thing, but in this view, here we're in G1. So cyclins, CDKs 4 and 6, along with will get you up to the restriction point. And then to get through that restriction point, you need cyclin E and CDK2. So that gets you through that restriction point into S phase. Once you're in S phase, cyclin E gets degraded and cyclin A takes over onto CDK2. And that will get you halfway through S phase. And then here in the middle of S phase, cyclin A is going to jump off CDK2, jump on CDC2, and that gets you into and most of the way through G2. And then now we're back to our first lectures with cyclin B and CDC2 to get you through that last transition. And then once you get through that last transition, you get degradation of cyclin B and you're back into G1. So keep in mind too that as you're going through, cyclin E gets degraded and that makes it go away. And so you can't go backwards in the cell cycle. You can't stay at this point because you've got cyclin E and CDK2 because you degraded cyclin E. So cyclin A comes on, that pushes you forward in the cell cycle. And you can see that here. So every time that cyclin's role is done, it gets degraded. And that's typically ubiquitin mediated degradation. CUL1 being our important E1 ubiquitin ligase. The only cyclin that isn't controlled by its degradation is cyclin D. Cyclin D is key here, very important proto-oncogene because it's the only thing that's going to be controlled by transcription, or not transcription factors, hormones. So hormones are controlling nuclear D, and that has you go into past the R point. From there on, the cell's no longer responsive to growth factors. It's just this internal clock. It's like the DNA, the program in the DNA plays out. And either the cell continues through to mitosis and divides, or it dies if something's wrong. Okay, questions about that?
You said it was cyclin B or D?
Cyclin D. D, yeah. And if we back up a tad, you'll see cyclin D is one of our key DEGs. Okay. So That is our cyclin and CDK story. Part of it anyway. The other part are CDK inhibitors. So we've got two classes of CDK inhibitors. You've got your INKs and your KIPs, KIP-4/6. So these bad boys, these are going to stop the cell in G1, right? This is responsible for getting us up to R. So this is going to stop us in G1. These stop everywhere else in the cycle. They'll stop the passage into S and the passage through the cell cycle after S. So these are two different kinds. And now what's kind of complicated and interesting here is these two are inhibitors of these, but they're activators of these over here. So when P21 binds to cyclin D and CDK4 or 6, it activates it. P27 also activates this, but it will inhibit these. And that becomes an important mechanism in controlling passage through the R restriction point. Because at the beginning in early G1, we've got lots of CDK inhibitors, and these are going to bind to the CDKs and CDKE and cyclin E, CDK2 are going to be inhibited. And it's activating these guys. So as you go into mid-late, now you're producing more and more cyclin D. As more cyclin D appears, it starts to bind up the CDK4 and again it's activated. And now you've got less and less spare P21 and P27 around to bind up the E CDK2. And as you increase cyclin D even further, then the cyclin D CDK4 binds up all the available inhibitors, and that frees up cyclin E CDK2 to stimulate the cell to go forward in the cell cycle. So that's kind of an important mechanism there. It has to do with, again, transcription of DEGs, making more and more cyclin D until that outnumbers the cyclin E CDK2. But this cyclin D CDK4/6 also has a role in its own. So when it's activated, it's going to hypophosphorylate RB. So right at the end of mitosis, RB is going to be totally dephosphorylated. So no phosphate at all in early G1. As cyclin D builds up, it starts to get hypophosphorylated. And that's going to prime it for later phosphorylations, basically. But it is bound to E2 and blocking E2, which is blocking the restriction point. So basically, this is hypophosphorylating RB, getting you up to the restriction point. And then when we get to the point that we've got active cyclin E CDK2, that will hyperphosphorylate RB, and that causes it to let go of E2. So that's the key to getting through the restriction point, is when you've got some free cyclin E CDK2 and you've already hypophosphorylated RB, that's going to allow you to get through. Okay. And then here, this is really good. I can practically guarantee you'll get a question about this on the exam. This is two positive feedback loops that make this transition go quickly, okay? Because, you know, the cell, it's more efficient if the cell goes quickly into cell cycle rather than, you know, half in, half out. So we've got two different positive feedback loops. One is here, once you hyperphosphorylate RB, E2F is released, and E2F causes its own transcription, so you get more E2F. Eventually, you're going to have more E2F than there is hypophosphorylated RB, so that's going to get you through. But also, E2F is going to make more cyclin E, and cyclin E then increases hyperphosphorylation of RB. So really, we've got two positive feedback loops here, but that's only one of our exciting mechanisms here. The other one is this one, where cyclin E CDK2 will phosphorylate P27, and that makes it a target for our ubiquitin ligase CUL1. So P27 then gets broken down, and that releases this cyclin E CDK2. So now we've got more active cyclin E CDK2, phosphorylating more P27, breaking it down. So again, a positive feedback loop where as you activate cyclin E CDK2, you're releasing more cyclin E CDK2. So the concentration of P27 falls rapidly, and the concentration of E2F and cyclin E increase rapidly because of these positive feedback loops. Any questions about that? Okay.
What stops the cyclin E CDK2 complex from phosphorylating the P21 when it's earlier in the cycle? We're looking at like when there's the cyclin E.
Right, because here it's blocked by that P27, and there's no free cyclin E CDK2 available. Once it does become available, these are going to go back and phosphorylate them. Okay. Oh, here's our acetylation again. So I like to throw this out here because we talked about it with our, I believe it was the estrogen receptor in chapter 3, how deacetylation and acetylation of the histones is really important in controlling gene activation. So here in the hypophosphorylated RB form, this hypophosphorylated RB brings in histone deacetylase, and that takes the acetate groups off histones and allows the DNA to be compacted up. So it's not transcriptionally available. And then once it's hyperphosphorylated and it releases E2F, E2F then will bind histone acetylase and will open up the DNA, and then we transcribe. Here's another thing I didn't really mention, but it's important. E2F, you've got 3 different E2Fs. 3. That's not that important. But you've got DP here as well. So DP is always going to be the co-activator with E2F. So keep that in mind. Acylated DNA transcription ready. All right. And this is just a broad general thing. Just all roads lead to cyclin D, basically. We talked about growth factor receptors, RAF, MEK, cyclin D, RAL pathway ends up in cyclin D, AKT, peak protein kinase 3, ends up in beta-catenin, which then activates cyclin D, hedgehog, JAK-STAT, NF-kappa B, everything leads to cyclin D. So cyclin D is really the center of what gets you up to the restriction point and then through it. Okay, so this is something I talked about P53 last time, but I didn't talk about this, and I think it fits better here than with P53. So this is new, but I want to talk about it. So MYC is just such an amazing IEG. MYC alone can cause a cell to divide. It's such a strong IEG. In fact, I think I wrote it in the notes there that when they first did their induced pluripotent stem cells, this is back during the Bush administration when they weren't allowed to use embryonic stem cells. They were taking like skin cells and adding genes to them to make them pluripotent stem cells. And one of the genes they added was MYC, and that shows you just how powerful MYC is as a cell stimulation. Beta helix loop helix structure, the helix loop, and here you can see the alpha helices are reading the base pairs in the DNA. So this is an experiment that shows you just how powerful MYC is. So what they've done here is they've taken an estrogen receptor and made a chimera between the estrogen receptor and the MYC transcription factor. So the estrogen receptor, when estrogen is not around, is in the cytoplasm. And when estrogen is around, it activates the estrogen receptor to be transported into the nucleus where it acts as a transcription factor. So they're taking advantage of that activity of the estrogen receptor and linking it to MYC so they can see what MYC does. So what they see then is when they activate the MYC with ER, that alone is enough to get the cells through restriction point and activate cell division. So MYC, really, really important proto-oncogene. Another thing about MYC that we see here is its cofactor. One of its cofactors is MAX, and the other is MIS1. So we'll see that here. So MYC has got two different cofactors. Here we're only looking at MAX. So MYC and MAX together is a mitogenic signal. That's what's going to get us through the restriction point and cause cell division. But if you've got another protein around like MSD, it can bind to the cofactor of MYC, and that represses signals. So we're getting into here a really key thing in development when you take development, if you do, how cells can either be proliferative and dividing or post-mitotic. Okay, so they're not dividing anymore and differentiating into adult cells. So it's kind of a switch between proliferation and differentiation. So it depends on what is going to be binding to this MAX. MYC and MAX together, on the next page we can see, MYC and MAX together are going to activate production of DEGs, okay, cyclin D, CDK4. It's also going to activate CUL1. Remember, that's our ubiquitin ligase that's pushing us forward in the cell cycle. And we're going to activate production of E2F. So again, if you only have MYC and you don't have cyclin D and CDK4, you're going to make so much E2F that you won't have enough PRB and divide. So this is a very powerful stimulator of cell division. MYC can also bind to MIS1, and this is also promoting in that it is blocking P15 production. P15 is made by TGF-beta, we'll see that in a second, but it's blocking at our G1 site. And it's also blocking the other CDKs, the KIP version of CDKs. And by blocking these, they can no longer block cyclin E, and so that's causing RB to be activated. I mean, RB to be blocked and E2F to be released. Does that make sense? Kind of. You probably need to go over it a couple of times. Basically, all I'm trying to say here is MYC causes cell division, and it causes that by binding to MAX, which is giving us DEGs and CUL1. And it does that by activating, binding to MIS and blocking the CDKIs. Okay. And this whole business over here leads us back to what we talked about before with activation of cyclin E and CDK2. All right. Are we good with that? Alpha flag, yes? Okay. All right. Now, TGF-beta, transforming growth factor beta. The interesting thing about this hormone is it tends to repress cell cycle and not activate cell cycle. It's called growth factor, but sometimes it depends on where it is, really, whether it's going to stimulate growth or repress it. Depends on what the cells are and how they respond. But anyway, this is how TGF-beta represses the cell cycle. So we talked before about our receptor SMADs, SMADs 2 and 3, then binds to the cellular SMAD, SMAD4, and that makes a heterotrimer here. That's going to bind to MIS. Now you remember up here, MIS is blocking MIS is blocking production of CDKIs. Here, this is going to cause the transcription of CDKIs, especially P15. And P15 is going to block us in G1. So keep in mind TGF-beta, P15 blocking in G1. The activated SMAD will also bind to E2F instead of DP, and that's going to be a repression. So that's going to repress production of MYC. So TGF-beta is directly repressing MYC production and directly activating CDKI production, and that's blocking cell cycle. Another thing I've added into the side over here is how MYC and TGF beta kind of fight it out. In that TGF beta is blocking MYC, but MYC, when this is around, blocking the CDKIs. Like we saw that in the last slide. So they kind of, another kind of physiological antagonism. But if you've got TGF beta around, it's upstream of making MYC. So TGF beta is going to win, basically. It's going to block the cell cycle. Okay. CUL1, I want to talk a little bit about this here because it comes up over and over again. It's basically moving the cell forward in the cell cycle. So these are just little snippets from previous slides. Remember that one of our positive feedback loops here was that cyclin E is going to phosphorylate P27, and that causes it to be degraded. So that's CUL1 again, helping us move forward in cell cycle. And this cyclin E degradation is CUL1 mediated as well. There's another CUL1 mechanism. Oh, it also ubiquitinates any free E2F that's sitting around. So that's another way it's moving itself forward, right? After you get through the restriction point, you get rid of E2F, and then the cell continues on in S phase. So we've got three different ways here that CUL1 is going to keep you moving forward in the cell cycle. I hate to say it, but that sounds like an exam question too. Okay. P53. So we've talked about this before. So we did these notes. I'm going to go a little bit fast. I hope that's okay. Because I still have apoptosis I want to talk about. Although we can do it Thursday if we run out of time. Okay. So P53 is the center of controlling cell division when there is DNA damage. And this is going to protect the cell from becoming cancerous, because when cancer is all about DNA damage. So if the cell can't protect itself, it gets more and more DNA damage. So here's just showing you what happens without P53. These are mice that are normal mice. Okay, they live, at least 500 days. Knock out one copy of P53, you lose some of your viability. Knock them both out, and these mice die pretty quickly. So P53 is really important. And P53 is going to be detecting, or kind of the nexus point of the detection of UV radiation damage, ionizing radiation, oncogenic signaling, and some of these other things. This is mainly what we're focused on, the DNA damage and oncogenic signaling. And here's an example that shows you how P53 helps you when there's DNA damage. These are normal cells. They're blasted with enough UV to kill them. Here's green. This is normal, positive control. That much UV kills them. You've only got one P53. It survives a little bit better, but still dies. You got no P53. They don't really care. They're doing fine, even though they've been blasted. Which doesn't mean they're healthy. It means they're unhealthy because they're continuing to divide with massive DNA damage. Okay, and this is how that system works. MDM2 over here is again an E3 ubiquitin ligase, and it is constantly phosphorylating, ubiquitinating P53 and degrading it. So just like beta-catenin, on a tonic basis, P53 is being degraded. So normally that's happening. When there's DNA damage, that activates ATM or ATR. ATM is looking for double-strand breaks. ATR is looking for single-strand breaks. When they get activated, they activate our checkpoint kinases, check 1 and check 2. And those checkpoint kinases then phosphorylate P53. And that protects it. P53 now is no longer a substrate for MDM2, and it builds up rapidly in the cell. So here's a question I've had before. Why do this? Why not have a system where when there's DNA damage, you activate production of P53? And the thing is, it's just a whole lot faster this way than the other way. So to increase the concentration of P53, if you had to increase transcription, it'd be like transcribe 30 minutes, transport out of the nucleus, translate another 30 minutes, it'd be 60 minutes before it gets back in the nucleus. Here, you just stop breaking it down and immediately it's coming up, okay? So it's really a really quick way to stop the cell when there's serious damage. And then what happens here depends on how much damage there is. If there's just a certain amount of, a small amount of damage, we're gonna transcribe P21. We're also going to be transcribing DNA repair enzymes, okay? So we're gonna block the cell from dividing, and we're going to increase DNA repair and fix the damage. Once the damage is all fixed, we degrade P53 and the cell cycle continues on its way. But if you've got a lot of damage, P53 activates PUMA, and PUMA is a pro-apoptotic BCL2 protein. So that inhibits our anti-apoptotic BCL2, and the cell goes ahead and does apoptosis and dies. So A or B, if there's small damage, we stop the cell cycle and repair it. A lot of damage, we go ahead and cause the cell to die.
How is it controlled whether the P53 does the DNA damage versus
Yeah.
like well doing gene expression like NER or MER would do it? Like if we
You mean how do we tell if it's going to go on this pathway or this pathway? Yeah, I don't know. Good question. I always assume it's about the amount of protection. Could be about the amount of phosphorylation. I don't really know that one. Sorry. You found the one thing I don't know out of my class. Just kidding. Good question. Okay. And this isn't that important, but it's kind of interesting how MDM2 and P53 regulate each other's concentrations. Let's see, this is P53. So MDM2 is going to increment P53 and it goes down. And then because P53 causes MDM2 transcription and it's low, MDM2 goes down. MDM2 is low, and then P53 comes up. Now it transcribes more MDM2, get more MDM2, and P53 gets broken down. So they kind of regulate each other. Not that important, but it's kind of interesting. Okay, now this is important. So P53 is a tetramer. And what they're trying to say here is if you've got, let's say you've got two genes, right? Everybody's got two genes for everything. So we've got two genes for P53. And if the cell's going to make P53 from both genes. If you've got one of your genes has got a point mutation, a specific point mutation inactivates that P53. Both genes are going to be transcribed and translated, and the proteins assort randomly. So randomly, if you've got tetramers, that means you've got 2 to the 4th combinations of possibilities of these subunits getting together. And if you've got even one of those point mutated P53s, it inactivates the whole complex. So that's how P53 can act as a dominant negative. One of your two genes is making a mutant form, and both forms get combined randomly. And so just by the statistical randomness, only one of your 16 complexes can be active. And so what that means is then a point mutation is worse than losing the whole gene. So we've seen this before. Here's your normal P53 wild type. You knock out one of these wild types Now you've got P53 with the normal and none. So the cell can still make protein from the wild type, and so it acts almost normally. Lower level of P53 because you only got one gene making it, but it can act almost normally. So you can see that here. This is normal. This is lacking one gene. Now you've got a point mutation, and it acts as a dominant negative. So now it's going to knock out 15/16 of the activity of the one wild type gene. And so you get almost no function, and you end up with this. Okay, so that's kind of interesting in that you can have cancers where you've got P53, and actually P53 is really high, and the cells aren't dying. Why is that? Well, it's because of this point mutation kind of a problem here. Okay. Okay. Now we're switching gears. So that was P53 and the P53 story. Now we want to talk about how if a cell is overstimulated, like say it this can happen, like you've got the gene for E2F on one chromosome and you get a chromosomal rearrangement, and that ends up on a different chromosome and it ends up in an active area of that different chromosome. Talk about cancer mechanisms in the next chapter, but so suddenly you're making way too much E2F. So you've got tons of E2F in the cell. That cell now it's oncogenic, right? That's converting that proto-oncogene E2F into an oncogene. Now it's overexpressed and the cell's dividing a lot. So what happens then is the cell goes into apoptosis. And this is an explanation of why that happens. So here is another experiment with our estrogen receptor. This time we've got E2F as the transcription factor. So when you don't have estrogen, it's sequestered in the cytoplasm. Estrogen goes in the nucleus. So here we are with these cells. This is normally without estrogen. And this is a cell sorter. So you're counting cell size. And all our cells fit this nice bell-shaped curve here around a fluorescence of 100. You then stimulate with estrogen, and you can see this peaks out at about, 575. Now our peak is down to 2.25, and we've got all these pieces of cells that are less than the size of a normal cell. So by fluorescence intensity, you're talking about cell size. So now we've got a whole bunch of chunks of cells that are fluorescing, that so 32% of our cells then are less than full. These are apoptotic bodies. So what we're seeing here is when you stimulate the cell, overstimulate the cell with E2F, that causes it to go into apoptosis. Okay, that makes sense? All right, why is that? This is a gorgeous mechanism. I just love this. So there's a gene that has got two different reading frames on the same strand. It's not even backwards, it's on the same strand. It's called, and one protein is called ARF because it's from an alternate reading frame. So we talked about P16. It's one of the INK type PDK inhibitors, CKI inhibitors, it's a CDKI. I'm getting confused here. So it's an inhibitor of CDKs 4 and 6, keeping you in G1. So that's your P16. You've got 1, 2, 3 different exons, and they're stitched together in this way. The red is your open reading frame, and so that is the protein. This is 3' non-translated. Now over here is a different promoter, and this promoter is going to activate transcription of this exon, and this exon. So these two proteins are going to share an exon. And so this is the reading frame of P14, and this is the reading frame of P16. So P14 then is a protein made from similar gene here, almost the same gene, and it's going to inhibit MDM2. So this P14 promoter is going to be activated when you've got an excess of E2F around. When E2F is like over the charts, we activate P14. Okay, so here's normal MDM2. So MDM2 is ubiquitinating P53 and it's getting degraded. This is normal over here. Now, when you've got too much E2F, E2F then causes P14 to be made, and that makes ARF. ARF then grabs MDM2 and sequesters it in the nucleolus. For a buck, what does the nucleolus normally do? What's the role of the nucleolus?
DNA replication.
What?
DNA replication.
No, not DNA replication. The nucleolus is where the ribosomes are made. Isn't that weird? It's like the only thing I know about the nucleus is that, except for this. And this is a new role for your nucleolus. You guys know what nucleolus is, right? Dark area in the nucleus. Nucleolus, a little nucleus, basically. So anyway, bottom line, when you've got too much E2F around, that makes E2F. It sequesters MDM2 in the nucleolus. P53 accumulates, and the cell goes into apoptosis, right? Activating transcription of PUMA. Okay, and here's kind of a different way of looking at it. E2F activates RF. ARF blocks MDM2, which was blocking P53. It's no longer blocking P53, and we get apoptosis.
So like too much P53 causes apoptosis, but didn't you say it was involved in the repair mechanism?
Yeah, that's.
So what changed here that caused it?
It depends on how stimulated P53 is. If you look down, way down the bottom here, if it builds up to a certain level, you're getting cell cycle arrest. Builds up to a higher level, you're getting apoptosis. Okay.
This is concentration.
I believe so. I believe so. Yeah.
I got one right.
Okay. Okay, and that brings us to apoptosis. So these are cells, this is a normal epithelial sheet, flat cells. And these are epithelial cells going into apoptosis. They round up and you can see membrane blebs, these big balloons, kind of like an aneurysm in an artery, right? Just blebbing out. Or, you know, if you've got a balloon and there's one spot that's weakened, it can blow out like that. Those are blebs. So there we are. Apoptosis. So apoptosis involves basically 4 steps, although it's hard really dividing into different steps, but I don't have notes here. Oh, I not have notes. I don't have my notes here, so I can't read them. All right, I'm gonna have to make this up. I'm making all this up, by the way. None of this is true. So what happens? The cell gets stimulated going to apoptosis, and it activates proteases. So these proteases are going to go out and break down the cytoskeleton, and that's going to make the membrane start to bleb out. Remember, membranes are not happy in the form that they are in a cell, right? They need to be held in place. Lipid bilayer will collapse into vesicles if you allow it. So you start breaking down the cytoskeleton, and the cell structure starts to turn into blebs like this. Also, you're activating DNases, and the DNases will go into the nucleus and break the DNA down into 200 base pair fragments, basically. And then eventually the cell breaks up into chunks, these apoptotic bodies, and then phagocytotic cells will come and clean them up. So these apoptotic bodies here that the cell falls into are what we were seeing here. Oops. Here, right? Chunks of cells. So apoptosis then involves the cytoskeleton breaking down, the mitochondria break down, and cell falls apart into chunks, and then phagocytes come in and take care of it. Now, why does this happen? This is a normal mechanism for, especially in development, you see it a lot. Just examples in development are the tail. You know, we all had a tail when we were embryos, and that tail dies back for most people. That's apoptosis. The webbing between your fingers, right? don't have webbing between our fingers, but as embryos we do, and those cells die back by apoptosis. So there's a lot of body remodeling that happens to apoptosis. Also, breasts, right? When a woman gets pregnant, her breasts enlarge and they get ready for lactation. After weaning, they die back, the cells die back and the breast gets smaller again. That's apoptosis. So these are just normal mechanisms of apoptosis. And also imagine you can call it normal when a cell is toxic and it dies, right? So it's being directed to die either by other cells or by internal mechanisms. And that's what we want to talk about here are extrinsic mechanisms and intrinsic mechanisms that turn on this apoptosis in cells.
Quickly.
Yep.
Just the apoptotic body ever get recycled, like some of the things that are in there, will that be like mitochondria? Like,
I believe so. I think after phagocytosis, the cell will fuse, what do you call it? Oh God, what do you call it? The pre-lysosomes, the endosomes. They fuse endosomes to their endocytosed material, and then that vesicle will acidify and become a lysosome, break it all down to monomers, okay? So it's not going to take a mitochondria and reuse it, but it's going to take all the pieces, break them down to monomers, amino acids, fats, sugars, and reuse those for cell growth.
what is the thing from the cell that gets completely destroyed rather than turned into monomers and reused? But what is something that gets completely gone?
Well, I imagine some of the monomers are just going to be eaten, right? Amino acids can go into metabolism and be broken down. So if the phagocyte or the phagocytotic cell doesn't need any more amino acids, it'll probably just use it for a guess. I don't know. I'm getting myself. I think, the thing about making stuff up, I say this a lot, I warn you when I'm doing it. When I know what I'm saying, I'll tell you I know what I'm saying. But this is kind of what I'm looking for you in those written questions at the end of the exam. Hypothesis. What do you think causes this to happen? Based on what you know, where do you think this is going? So that's what I mean when I'm making it up. I mean, phagocyte, the cell takes up these apoptotic bodies. So what do we know about phagocytosis? They then fuse with your endosomes and you make lysosomes, you break it down. I said back here too that the DNA gets broken up into 200 base pair fragments. The reason I throw this figure in is just to show you, this is the nucleome structure with the histones wrapping around the DNA. And between here and here is 200 bases. So when the DNA gets activated, it just starts cleaving. And so you get this ladder of fragments of genomic DNA that are 200 base pair increments based on that kind of random digestion of the DNA. Not that important, but I think it's interesting. It's one of the interesting things about this. Okay. All right. So what is the mechanism? How does this happen? It comes down to two different things. One are the caspases. So the caspases are the proteases that are going to go out and do all the destruction in the cell. And we've got two different kinds of caspases. We've got initiators and executioners. So your executioners mainly caspase 3. So they're called caspases because they've got the C is cysteine. They've got a cysteine in their active site. And aspartate, they're going to go out and cleave anything with aspartate in it, which is basically cytoskeleton. You've got aspartates everywhere. So caspase 3 then is an enzyme that's going to go out, digest the cytoskeleton, make the whole cell collapse. These initiator caspases are going to activate caspase 3. So these get activated by different mechanisms. Caspase 8 by the extrinsic, caspase 9 by the intrinsic mechanism, and they activate caspase 3, and then the cell collapses. Now, all of these caspases are stored in the cell as procaspases. So there's a peptide that's across the active site that stops them from being activated. And imagine this, every cell in your body right now could explode in the next 30 minutes. You would be just jello. Isn't that crazy? I should go to work for the military and develop new chemical weapons. Anyway, the other part of the story are the BCL2 proteins. So I guess we should start at the bottom. If you release cytochrome C from the mitochondria, then cytochrome C will come out and bind to APAF1 and then procaspase 9. And it all comes together to form this huge complex called an apoptosome. And that caspase 9 then gets activated, and that activates caspase 2. So key here being cytochrome C. So anti-apoptotic BCL2 proteins are blocking the release of cytochrome C from the mitochondria. And we really only have two, BCL2, which is kind of the eponymous factor here of the whole group, and then BCL-XL. So these are your pro-apoptotic BCL2s, or your anti-apoptotic. So they're blocking cytochrome C. These are going to promote cytochrome C release. And we've talked about some of these already. BIM we've talked about, if you remember back to AKT, phosphorylates FOXO, which stops BIM production. AKT directly phosphorylates BAD and inhibits it. BID is another one. BIC, PUMA we talked about, increasing P52 causes production of PUMA. All of these things are basically like a stoichiometrically inhibiting BCL2 and BCL2X. So these are sitting there blocking a pore in the mitochondria, stopping the release of cytochrome C. When these come along, they bind to these and block their activity, and that allows cytochrome C to be released. So it's a stoichiometric kind of process. If you've got more of these, then they win and you get apoptosis. If you've got more of these, win and the cell survives. Keep that in mind in terms of cancer too, because this can become an oncogene by being overproduced. You've got two, the cell can't go into apoptosis when it should, and it becomes cancerous. But anyway, back to our story here. It's all about cytochrome C stored in the nucleus. Okay, so here's our apoptosome. Cytochrome C comes out, it binds to APAF1, which then binds to other APAF1s, and that recruits procaspase 9 in the center here. And procaspase 9 then activates, and it will activate caspase 3. And here's what it looks like. We call it the wheel of death. Okay, so our two pathways now. I hope I didn't go through this too quickly. One is the extrinsic pathway. So the extrinsic pathway has to do with some cell, like a cytotoxic lymphocyte, telling a cell to die. So this is like, your immune system's looking around for sick cells, and when it finds a sick cell, it tells that cell to die through apoptosis. And it does that with a protein in its membrane called Fas ligand. The receptor then is Fas. And this is that juxtacrine hormone stimulation, where the ligand's attached to one cell, the receptor's attached to another cell. So this cell touches that cell, tells it to die. So we've got a adaptor protein here called Fas-associated death domain, FAD, F-A-D-D, Fas-associated death domain. So FADD then binds to procaspase 8, and when it's stimulated by Fas ligand, that activates it to become caspase 8. Caspase 8 then activates caspase 3, and you get activation of apoptosis. Caspase 8 is also going to activate a BCL2 protein. I believe it's BIM. I don't remember exactly, but it also stimulates the other pathway as well. So that's your extrinsic pathway. Now, I've been harping on this since the beginning of the class, I think. The whole idea that cells have to be attached to something to stay alive came up first in chapter 3 when we're talking about cell crawling and integrins and focal adhesions. Remember that whole story? And that focal adhesion complex includes a number of kinases. It's got focal adhesion kinase and SARC. And that's gonna phosphorylate and cause SOS, GRAB2, RAS, RAF, MEC, MAP, all that stuff. So when the integrins are attached to extracellular matrix protein, fibronectin, activates cell signaling, and that keeps the cell alive. And that's called trophic factors or survival factors. When that lets go, so say you take a piece of tissue and hit it with collagenase, destroy all the extracellular matrix proteins, put them in solution, they're no longer holding anything, they're gonna die. Cells will go into apoptosis when that happens. And that's because when you don't have those survival factors, that's going to increase the production of BCL2s. Remember, we said you activate AKT, you're inhibiting BAD. So now we're not inhibiting BAD anymore, and it's building up in the cell. And pro-apoptotic BCL2s, when they outnumber our anti-apoptotic BCL2, cytochrome C release, production of the apoptosome. Okay, so that's one intrinsic pathway. And that, when we get into cancer, we'll talk about that's why normally cells don't crawl away and go off and become metastatic, because as soon as they get out of that tissue, they die. So that's a protective mechanism. And then we get back to, oh, and then caspase 9 activates caspase 3. And again, we're breaking down. And then our P53 mechanism, right? When P53 builds up, we get PUMA, and PUMA just activates as a pro-apoptotic BCL2 and cytochrome C release. Okay. I want to show you an animation, because this is a really cool animation. I mean, I have this in your site. This is so those yellow things are fast. You see fast ligand coming in here. I believe this is a mitochondria. That's sort of close. So the cell rounds up as it uses the integrity of the plasma. That, okay. That's pretty cool, huh? Okay, any questions? All right, that's it for this exam. It's an awful lot of material. We will, like I said, I will get you an exam today to look at, and we'll have a review on Thursday. Then you'll all ace the exam. That's all there is to it. Any questions, comments?
Hi, Dr. Sliss. I just wanted to double check. So that means that the exam material ends there at like slide 69 or 70?
Right, if you saved a PowerPoint previously, you should get rid of that one because I deleted that one and I uploaded a new one.
Yeah, I saw you uploaded a new one this morning, right?
Right.
Okay.
And
Yep.
that new one will stop where we stop.
Okay. All right. Sounds great. Thank you.
Okay. Any other questions from the crowd? David, Isabel, sorry. Isabel, can I share your news? Or
Yeah, you can share my news.
she got into dental school.
Yeah.
Yay, Isabel.
Congrats.
Thank you, everyone. I'm very excited. Moving to Utah in June.
Her dad's my dentist, by the way. So I'm paying for her education. I hope you appreciate it.
Thanks,
All right,
Dr.
that's all
Slish.
I got. That's all I got for today, okay? All right, I'm gonna let y'all go.